be presenting this workshop, and he's actually an instructor with Biotech Without Borders, and he's calling in from his own home with his microscopes in his own lab, I believe. Is that right, Craig?
Yeah, a little impromptu home lab. So it's fun turning 30 and moving back in with your parents.
Yeah. All right, Craig, take it away.
All righty. So we'll go ahead and start by sharing my screen. All right. Well, thank you everyone for showing up. We're going to start the session just with a bit of a presentation. We're going to kind of go over a bit of the details. If anyone was at my talk yesterday, lightning talk about soil ecology, I'm going to cover some basics, but we're going to kind of go over the cast of characters. We'll probably try to see and kind of talk about like the benefits of using direct soil microscopy versus like doing colony forming units, so like a plate counts. Anyway, so this is my nice little setup here. I got a little USB coin microscope, dissection microscope, got a compound light microscope that's got a little USB camera in it. So we'll be switching back and forth. But for the start of this, we're just going to get into the slides and we'll go from there. So since we have the full hour and the slides are mostly pictures, if you guys want to ask questions, I'm going to stop twice during the first presentation part and we can ask some questions about the new information. And then once we get into the actual, once we actually get into the soil microscopy component, we might do the same in general, but we're going to kind of keep it nice and easy and free flowing. But yeah, all righty, here we go. So what is soil? That's a great question. It's really interesting because a lot of people think, you know, they don't really think much about it. We actually know, like, you know, there's a famous Leonardo da Vinci quote that we know more about the planetary bodies moving above the cosmos than we know about the soil beneath our feet. And it's something that's kind of this black box throughout history that's been super important. Anyway, so Hans Jenny, one of the founders of modern soil science, basically defined it as three parts: mineral, the sand, silt, and clay, the parent material, which pretty much is degraded by weathering, by abiotic factors, and then the biological weathering of the biotic factors. Organic matter, humus, detritus, organic matter that's being decomposing or exudates that organisms put out. In nature, nothing is wasted. One organism's waste is another organism's food. And then the biology, the microecology, the trillions and trillions of microbes that are having these nesting ecologies that pretty much build and allow, you know, life to exist on a macroecology scale. So this is probably a diagram most people are familiar with when they hear about soil, learn about it. This is the soil texture triangle. So it talks about, you know, based upon what proportion of whether you live in an area where your soil is sandier, siltier, or clayer, whether you have a propensity for to have high porosity or low porosity, good drainage, low drainage is based upon it as well. And so this is a good starting point, but the reality is most soil science kind of stops here. They kind of see soil as an extension of you know, geology, which is true. You know, the geomorphology of your soil is really important, but they don't really pay too much to biology. So we're starting to realize how important the microbiology of our soils are for, you know, for the plants that we want to grow to eat, to basically have healthy environments if we have anthropogenic disturbance. And even further, you know, our own gut microbiomes. We're learning more and more about how our gut is very intimately tied to the soil. So this is what soil more actually looks like. You know, we're familiar with the horizon layers, but on the right, it's an ecosystem. It's one could even say it's a superorganism. It's a composite organism where all these assemblages of microbes are living together, having these complex systems, much like these different pathways within our own bodies with as higher eukaryotes in general. So it's really important we kind of understand these details and differentiations. But we see the basic example here of the different horizons. You know, the top horizon being the O horizon, then the OH, the O humus horizon, which is the organic matter that's been decomposed. So olmics, fulvics, and humic acids are these large molecular weight, these large molecular compounds that are enormous. They're tremendous and huge. And basically humic acids aren't extracted and processed from mineral ore, or actually produced by fungi. And even further are actually food sources that are, you know, exudates that fungi can solubilize. So the huge part of our topsoil is composed mostly of these humates produced by fungi, that the more complex molecules that bacteria can't really break down. And we go down to the surface, the subsoil, the substratum, and we're realizing that life exists far down into the substratum, even parent material. We're finding nematodes far down as several kilometers into the Earth's surface, and we're even realizing that there's the deep ecology. The Deep Carbon Observatory, I think back in 2018, realized that there's even more life that evolved in this kind of on a frame of geomorphology than the life that is on top of the Earth, that actually is used to this diurnal cycle of feeding off the sun's energy gradient. So it's pretty amazing. So we're only living on just the surface, the skin of the Earth, but it is indeed the flesh of the Earth. So how does soil get formed? We tend to think about our lives or our places we live in an ecological time frame, but we understand this is all underpinned by the geological time frames. If anyone's ever seen images of supercontinents, Gowanda or Pangea, these are old time frames which these tectonics and geomorphologies are happening. And we have a disturbance event, either a fire, human activity, a tidal wave. This is something that's super common. And this is actually true for most of the microorganisms because they've been through it all. Billions of years of these disturbance events. So what happens if you have a disturbance event? You kind of get the reset, the ecological reset back to the parent material. So you have the first kind of species, usually lichens, algae, forming these assemblages. We hypothesize that the first organisms actually moved down to land over 500 million years ago, were these assemblages of fungi and algae forming these lichens and the ancestors to basically modern land plants. So over time, you get a buildup of organic matter. You get these early kind of crusts and lichens, then you get some of the liverworts and kind of these ancient kind of sporulating plants, and then you start getting the herbaceous plants. Then you start getting into more of the plants in general. And what happens is you're actually increasing the fungal biodiversity. So what's happening in general, fungi have been here for a while, but also the decomposer fungi, the saprophytic fungi. So here's a nice little example. This is a great textbook, Soil Morphology, Ecology, and Biochemistry. I'll be putting this in a document. I'm going to upload it later. But it basically goes to the aspect that you have the abiotic, which is the weather and wind erosion, and then the abiotic, and then you have the biotic, which is living organisms in general. So this builds topsoil, and you actually have the humal layer. However, through anthropogenic activity, you have overtilling and even just simple for agricultural practices. Most civilizations, human civilizations, pretty much fell apart because they mismanaged their soils, because it allowed them, their population to grow, but then they basically climaxed and then couldn't support it as well. We know that the Romans tilled their soil 8 to 10 times a year. They tilled most of their topsoil straight into the Mediterranean Sea. So it's pretty important that we want to understand, where is our food being grown? What's the state of the ecology in general? We understand that soil cleans and filters water. We want to understand the microbiology, the organisms that are actually living in this ecosystem. So here's a great little photo. This is something that we'd actually call dirt. So this is an image of a soil microscopy slide, actually from Brooklyn, New York, in Greenpoint. And so this is right along the Gowanus Canal. It's the estuary that basically shoots off the East River that actually separates Queens and Brooklyn. So this is a super fun site. There are 6 to 7 layers of black mayonnaise in the bottom from all these petroleum refineries. And we can very easily say that these soils have gone through an ecological disturbance, an anthropogenic ecological disturbance. So what's really interesting is that there's all this compaction. There's a video we'll play later, and you'll actually see that this is all bacterially dominant, reminiscent of the initial disturbance event, where the bacteria are the ones that are basically able to solubilize the nutrients out of the parent material, kind of these lithotrophs, and then whatever organic materials left over from decomposing microbial bodies, higher organisms like ciliates, basically breaking this down in general. So this is an example of dirt. So let's understand that these words are not synonymous. They're very different. Basically in general, dirt is really just the parent material. It's just the sand, silt, and clay. What makes soil is the organic matter and the microbiology. And then based upon what types of plants will grow is depending upon what ratios of bacteria to fungi. And we'll often hear terms fungal dominant or bacterial dominant. In the most productive ecosystems, these old growth forest ecosystems, you have predominantly fungal dominant microecologies, but they're not dominant in the sense that they're the only organisms living in it. they're more so facilitating. They're the architects of the soil. They build the structure by which all this other microbiology, this microbial diversity is able to thrive and flourish in general. So we understand with soil, we get clean water. It's able to filter and store. We think about all of that, all those particulate areas where water is able to basically invoke its paramagnetism and bond to with its polar bonds and hold on to it as well, versus dirt, it'll just really run right through it. If you have a really high clay soil, it'll pool and it'll go anaerobic and it'll be super reduced. Or if you have a really sandy soil, that will just run straight off. And this is a huge issue because, you know, in the anthropogenic use of fertilizers and pesticides, we have pretty much the estuaries that feed into the Mississippi River are constantly pumped full there. We actually, it's known as Cancer Alley, the area in Louisiana that feeds off into the Gulf of Mexico. So it's very important we understand how we can basically have responsible management and practices of land and soil, you know, not just for growing food that's more nutritious and healthier for us, but also too that we manage the whole span of ecologies that are affected by this activity that ultimately is controlled by soil. So, you know, usually the way you look at soil microbes, or just the microbes in general, is you'll do a factor of colony forming units. And this kind of fits very nicely. This is a picture from Molecular Biology, the Cell 6th edition, and basically admits that, you know, in general, that the vast majority of microbes that are out there, you know, 99% of prokaryote species, these we've yet to sequence, because the reality, they're living on a variety of food sources that aren't, you know, dictated by an agar media. So the factor in general is that if we're looking at the methodology of taking a soil sample, putting it onto a petri dish, either with tryptone or potato dextrose to attract fungi or bacteria, we're often making this anaerobic. We're actually often sieving our soils and drying our soils. We're shredding all the microecology that's there. So an effective way to look at it is by doing live microscopy. This was really kind of pioneered by Dr. Elaine and a team of microecologists. You had nematologists, microbiologists study bacteria, you had mycologists, you had nematologists that understood all these different roles, that you have an ecosystem, a whole food web beneath the soil. And when we think about conservation and biodiversity, we often think of the rhinos, the pandas, the giraffes, which are very important animals, especially culturally and for the sake of understanding our place in ecology. But the microbiology, the ecology beneath their feet, that may be even more important to protect because it supports all the higher ecologies. So here we are, we have the soil food web. The soil food web is, very simply put, a mapping of organisms based upon their trophic levels. And we understand that we have these emergent capacities by which organisms will organize based upon where they're sitting, based upon the energy gradient that's basically ultimately coming from the sun. So we'll stop here. We'll take a couple questions, maybe like 3 questions. And does anyone have anyone? You could feel free to unmute your mic or raise your hand. If you have chat monitors, we can kind of go from there.
Craig, there's one question that you mentioned before, and I didn't get the words you said when you were explaining the difference between soil and dirt. And yeah. And you were saying a ratio between something and something, and I missed the word.
So dirt, as we call it, it's just the parent material. It's the sand, silt, and clay. It's just the literal mineral composition. But soil has the composite of the organic matter, and then the microbiology, the organisms that are producing this organic matter are able to cycle it in general. So those are the key things. So dirt, if you've ever been in a big city and walked over a tree planter and it's like hard as concrete, that's dirt, because really only bacteria living there, very low organic matter content. Versus if you go into a forest, you feel your cushion under your feet. There's a whole microecology going on in that aspect because they're cycling the nutrients, the carbon cycle, the nutrient cycle, and then even playing important roles in the mineral cycle if you have a developed microecology. We'll do like one more question.
One question. I'm working on a project that's looking at microbiology in a marine environment.
Mm-hmm.
I'm wondering if you have any sense of how the 2 types of ecosystem compare.
Oh, very important. So we think about the factor that, you know, all life started in the sea. You know, we're pretty much just sacks of seawater when you think about it. You look at a little prokaryote, a bacteria, to a fungi. You know, we really are all of the same ratios of water and that capacity in general. So when we understand different ecologies between terrestrial based and aquatic ecologies, Imagine the edge effect. Think about riparian ecologies. You have a doubling of species because these organisms are spending different portions of the lifespans in general. So a lot of the techniques by which you can look at aquatic microbes in the microscope, you can apply to the soil microbes, but you have to resuspend them in a water solution, which I have this little kit here. We'll kind of go over the details for the workshop component in general. But for the sake of aquatic ecology, for microscopy, it's very similar, just to kind of taking a sample and putting onto a slide based upon how much dilution there is to the biological material and the organisms in the sample.
Okay, I'm sorry, I was thinking of the ecologies themselves. I mean,
so the ecologies themselves, well, it's as you get further away from water, those are organisms that have been able to survive in either kind of, based upon the hydrological cycle, right? Obviously, a desert ecology is very different. It's not dead because if you get a big rain event, everything flourishes. The microbes, especially. So the bacteria and the nematodes and the protozoa, they'll insist. They'll make this hydrophobic. But if you go closer to the water, they're far more. They have much easier access. They've evolved that capacity. So it's with aquatic ecosystems, it just depends upon how the hydrogeological system is in place in that local ecology.
Thank you.
Great, cool. All right, so we got a little cast of characters here. The sun is an amazing thing. It's we understand that the predecessors like the cyanobacteria and algae discovered a way to utilize the charged particles of photons coming down from the sun and then fix carbon dioxide of the atmosphere. So successful they caused a mass extinction event. But you get to the point now, but in general, the ancestors of those organisms became the modern land plants. So plants, besides from the material, their plant and leaf material and fruit material or germ material falling back down onto the ground, they're also putting out exudates. They're actually putting out nutrients that will attract bacteria and fungi. And what they're doing is they're actually trading these nutrients back and forth. Because, for example, plants are great at making sugars, but, you can't live off a diet of potato chips and soda alone. It's the fact of this mutual exchange where these bacteria and fungi have fungi have a huge surface area, bacteria everywhere, and they can modulate because they're microorganisms, their external environment much easier than their internal environment. So they can solubilize sand, silt, and clay. They can solubilize the macronutrients, the micronutrients, and trace elements out of the sand, silt, and clay. And they're exchanging it with the actual plants in general. So we understand this more and with fungi because they're facilitating these exchanges so far as well. Then further, this is important, but this exchange, the bacteria and fungi are going to want a good exchange rate and deal. So what happens is in general, since basically bacteria and fungi, more so bacteria, mostly little just sacs of proteins, their carbon to nitrogen ratio is pretty narrow. But you look at the predators, the heterotrophic level, the arthropods, nematodes, the protozoa, they're going to be eating those bacteria and fungi. And what happens is they have a much higher carbon to nitrogen ratio that they get built up to. They eat the organism mostly for their carbon. So what happens is those arthropods, nematodes, and protozoa, they poop out all those nutrients. So they poop out those nutrients that are actually in a bioavailable form that the plant can take up. And those exudates in general go on to be processed by other microorganisms. So what happens if the protozoa, nematodes, and arthropods eat all the bacteria and fungi? That's a problem. So you have the fourth trophic level, you have these larger predatory nematodes, you have these macroarthropods, and you start building a self-regulating micro to macro ecology. So we understand it's very important. This is actually what like a soil structure should look like. You see a whole cast of characters, a micro ecosystem. Between plant roots, between mycorrhizal fungi, between saphotrophic fungi, between bacteria, protozoa, microarthropods, nematodes, the whole ecosystem is there. So here's an example of what a healthy soil will look like under the microscope. We see lots of structure. We see that this isn't all compacted together. We see little nuggets of these humic, fulvic, and ulmic substances, these little kind of dark spots by which bacteria are actually basically clinging to. They make these alkaline glues and bind to them as in general. But then, much like a bridge, the individual blocks of the bridge, you have the fungi that are acting like these girders, pulling it all together, making this macro ecology. So we understand in general that there are nutrients in the soil. Nutrients are obviously in the sand, silt, parent material, and depending upon your soil, depending upon that parent material, a different cation exchange capacity, the ability for it basically to transmit those different ions. However, its organic matter is basically interwoven in two. These huge organic molecules produced by fungi, these humic acids. This is like one segment of a humic acid. They're enormous. And these are incredibly complex molecules that fungi actually can break down. So the factor in general is a lot of soils, you have these nutrients. If you don't have good biology, for example, in compost, you could have a really great looking compost, like nice 70% chocolate bar. But if you don't got the biology, if you don't got the fungi, especially the saprotrophic decomposer fungi, it's kind of dead in the water. So it's pretty essential that we cultivate the whole range of microorganisms to basically liberate a bunch of these nutrients that are basically stored away. Humic acid has these humates have a huge, enormous cation exchange capacity. All those different spots for different elements in the soil can bind to. And this is possible by fungi, the decomposer fungi basically playing this role. And we understand this kind of like when you get a soil test, like, okay, like you go in there and get a bit of your soil. And what they're doing is they're looking at the different pools of nutrients, the total, the exchangeable, and soluble. So total is if you're just taking everything out using a super, pretty prolific acid, these different strong extractant, and just kind of seeing what's in there in general. Exchangeable is basically talking about what's bound, what's actually sitting on or kind of tied up to the different surfaces of your soil. And then the soluble is actually just what's flowing in there in general. And most fertilizers, those are more in the soluble pool because they're salts that are basically a composition of a cation bound to an anion that are basically soluble in water. However, documented in sparks, more so when we're talking about sparks, we're talking about soil chemistry, we're talking about environmental soil chemistry, Don Sparks 2003, that when they've done studies and look at the general global composition of soils, the nutrient or the macro, and nutrient and trace element concentrations are roughly kind of, equitable, basically equal upon global distribution. So what's really happening is that you have the microbiology of the bacteria and fungi are able to use, are able to basically extract these micronutrients, macronutrients, and trace elements out of the parent material and solubilize it up, making it available for plants. And then from there, we have the whole process of the poop loop, where you have the microecology basically living off this whole exudate capacity based upon these energy streams. We'll stop for a couple questions. Does anyone have any questions, or we'll just go ahead? Cool, I think we're good, cool. So this is affectionately called the poop loop. So basically it talks about in general that, in addition to dead organic matter, you have these different exudates, and you have the decomposers, aerobic bacteria, going on basically the organic matter, but also it's the mineral cycle. You have these bacteria and fungi are able to solubilize these nutrients out of the sand, silt, and clay, and then the organic matter, especially these more complex humates that are produced by the fungi. So little cast of characters, plants, pretty self-explanatory. They're basically utilizing the energy gradient from the sun and producing exudates. So they're producing sugars, proteins, based lipids that are attracting these different aspects. And what's amazing, based upon the plant root system, they're producing all different sorts of exudates. They're attracting different types of microbes that it can solubilize out there as well. So it's incredibly dynamic, this role. We have bacteria, basically being the organisms that are able to break down organic matter, in an aerobic environment. In anaerobic environments, they go through fermentation and producing different alcohols. This is anaerobic bacteria are more so happening in aquatic ecology, especially when you have different silt and sediment layers in the bottom of a stream, a lake, or a pond. Then we have fungi, fungi being, really the architects of the soil, and our fungi have a whole number of different roles to them. You have the saprotrophic fungi that are breaking down, like the wrecking crews, they're breaking down the hard wooden material. Usually when trees are getting older, they're able to break down the complex, not only the cellulose, the hemicellulose, but also the lignin, basically the structure that makes wood, the rigidity, the structure that gives plant heterogeneous structure that allows plants to remain upright. But then beyond the actual other saprotrophic fungi that are basically growing on lesser materials, but also more compost species, you have the mutualistic fungi, the symbiotic fungi. So it's pretty amazing because we have the vascular, we have the arbuscular forming fungi, which are some of the oldest ancient fungi that basically form these lineages with algae, form these lichens, to even to some most decomposer fungi that actually converge to be these these ectomycorrhizal fungi, the fungi that basically are forming these nets on the outside of plant roots and forming this symbiosis. And what's really amazing is that even beyond that, we have interchanges of roles that are basically happening within the actual soil. That these fungi are actually acting as highways for different microbes. So this is pretty amazing. So we'll do a quick little video to highlight. So what we're seeing here is we're seeing the mycelial network. We see the cytoplasmic stream on the inside, but these little bits of Pseudomonas streaming along this little filament of water on the outer surface of the hyphae. And this is amazing because in general, these fungi are the architects of the soil, like these super highways. So in addition to transferring nutrients between plants within them, they're actually facilitating the microecologies and basically these movements of different microbes throughout the soil. This is incredibly important because as we understand in more advanced ecologies, the further the succession, the more fungi there are, and they're facilitating these relationships and connections. So it's kind of interesting at a certain point, these advanced ecologies, it becomes like a singular mycelial map. It's pretty amazing, especially in these older growth systems. So it's pretty wild. So we have the protozoa. So we have a variety of, from counterclockwise from the top left, we have our ciliates, we have our flagellates, we have our testate amoeba, we have our, oh, I forget that one. And we have also too our free living amoeba in general and other flagellates. And so these are really interesting because based upon the compositions of them, this will indicate whether our soil is kind of an anaerobic situation, more of a dirt, or more of an actual soil, more of an aerobic environment in general. Usually the ciliates show up in more anaerobic conditions. But these are the organisms going to play pivotal roles for feasting upon the different bacteria and certain types of fungi as well. Basically different morphologies. A great resource if you really want to go down the rabbit hole. So microbiology, ecology, and biochemistry by Paul from 2003. I'll put that in the notes after the presentation. And then nematodes. Nematodes are everywhere. They're super prolific. From basically feeding on plants, which most people are familiar with nematodes as pests, agricultural pests, but also too, nematodes play a huge role of different functions. Based upon their morphology, some of them are actual root feeders, but some are actually fungal feeders. Some of them are bacterial feeders. Some are actually predatory nematodes that actually will hunt the pathogenic, the pest nematodes that will cause basically blight on certain types of plants. So the more ecological diversity you have in the soil, the more these assemblages and niches actually form in the lower microecology. Then with the arthropods, basically in general, we have microarthropods or the early stages of larger macroarthropods. And this is actually when we're starting to move into the macroecology, when you're actually getting birds or earthworms or different kind of moles or voles that are all building upon this energy gradient based upon the microecology. So it's really interesting because we're actually starting to realize that a lot of our current agroecosystems are based upon this bacterially dominant system, either from overtilling our soil, destroying all that fungal biodiversity, or overusing pesticides, fungicides, nematicides that are all basically killing the microbiology. And so we're kind of dependent upon these fertilizers that basically continue to feed our plants. If you feed plants exclusively fertilizers, they won't put out those exudates. But after a certain time, you disturb the balance of the soil, the natural kind of immune system, the natural kind of microecology. It's a superorganism. And this is when you actually have to deal with the pests and actually use all those. So if we shift more towards a bacterial to fungal ratio that is more equal, we'll basically have a healthier ecology. We'll attract more biodiversity on the microecology and also on the macroecology. And what's interesting in general is when we have a distressed plant, a bacterial dominant soil, the plant is basically putting most of that energy, most of its exudates into the soil to try to attract the bacteria and the fungi, the microecology, and actually putting less of that energy into its actual shoots, its fruits, and its leaves. So you can actually increase plant biomass. And then we understand we want to have carbon sequestration, increasing plant biomass with good composting practices. You can build those humates, build that, or the OH horizon, build that topsoil even faster than we previously thought. So that's basically the soil food web in a nutshell. And the poop loop. So we're gonna break out into the actual workshop component of the session, which is good because we're about halfway through. So I've got a little setup right here about all the stuff that you can basically do. First things first, pretty important, you need a microscope. a compound microscope that has a iris diaphragm, so you can control the basic light balance, because we're going to be shadowing, because a lot of these microbes are transparent. So if it's all blown out, if you can't control the intensity of the light and can't do the iris diaphragm, it's going to be hard to see the microbes in general. Secondly, you're going to want some microscope slides. You're going to want some
Greg,
cover
you
slips.
want to unshare your screen?
Oh yeah, that's super important. Okay, so we'll kind of just walk you through the equipment in general. So we're going to want our microscope slides, obviously. We're going to want some cover slips. We're going to want some, some test tubes. You can use a glass of water in general as well. It just helps to understand what your volume to mass ratio is. Basically, if you have a lot of bacteria in your soil, you need to dilute it to really do counts, but we'll simplify it here. Little eyedropper, because this is good. We're going to actually make our slides. And then what's really important is when you're making these dilutions in the soil, you're going to want to use like a purified or spring water. If you use chlorinated water, you're going to kill all your microbes. So that's something too that, like works as well. When you're working with soil, if it's in your garden, it's going to be pretty fine. Maybe just wash your hands. But if you're doing it from a site that has human disturbance or urban area, maybe you want to wear some gloves, because it's really important to protect yourself, because you may not know what's going on. For the purpose of the workshop, I'm going to be going into my my parents' garden on their balcony, and we're going to basically look at these microbes overall. So to get myself ready, we got to use proper PPE. So I'll put on my gloves. That's the fun part. It's really hard to put on gloves when everyone's watching. It's like the one funny thing. So cool. All right. So how are we going to extract our soil sample? Like what is this super high-tech expensive device? I don't know, an apple core. Because we're going to basically take a little 3-inch or 6 centimeter little wedge, and then we're going to throw it into a super high-tech soil containment device, a little Ziploc bag, and then we're going to label it. So we're going to take a marker and write on the outside. Don't put a piece of paper and put it in, because if you put it in the piece of paper, you're basically adding food for the microbes. You're giving them that sugar, that cellulose, they'll break down in general. So it's really important, because if you go to take a sample and say, oh yeah, I'll remember where it is. No, you won't, because you'll forget. So, the difference between science and messing around is writing your data down. So that's really important. A little spoon, because it's going to help us understand how much we can put in. One nice thing about it, 1 milliliter of volume is roughly equatable to 1 gram. So gotta love the metric system. All right, so to do this, we're going to do a couple things. First things, I'm going to put in some wireless headphones. So hopefully y'all can hear me when I. Can you hear me? Okay, thumbs up. All right, sweet. And then we're gonna have a little fun, because I'm gonna share my screen, but I'm gonna use the internet. I'm gonna use my fancy dancy iPhone, which it's an iPhone SE, so it's it's fancy. It's still using quantum mechanics to do all this stuff. And then I'm going to basically share my screen, so you guys can see what I'm doing as I'm going out onto the balcony to do this in general. Cool. So right now, all I need is just my plastic bag and my apple core. So hopefully the lag isn't too bad. Oh, you're good. I'm actually, no, it's fine. All right, so we're out on the porch, and normally you do your site assessment. I can put some more details about how you'd actually pull from a plant, but we're going to basically take a sample from this little planter right here. On my video, I'll do my flash so you guys can see. All right, so we got our little apple core. I'm going to pull from over here. Fungi earlier this year. Let's do a little apple core. We're going to dig in. Get a little 3-inch little plug right there. Perfect. All right, so we got a nice little 3-inch little plug. And now we're going to take our plastic bag. I'm going to liberate the clod, push it through. So we got this super high-tech soil sampling and containment device. It's pretty amazing what high technology we're working with today. All right, so Turn my flashlight off. Oh, you guys are still watching me do the collection. This will be fun. So I'll wait till it catches up.
It's kind of like time travel. I think it's fantastic, really.
It's cool. It lets me prep it. Lets me prep the sample on the dissection scope, which is perfect. I was like, I really hope Zoom doesn't crash, and so when I tested it out, yeah. So anyway, but yeah, so I'm going in there. Yeah, pretty much I inoculated with some fungi earlier, some garden giants, some Stropharia rugosa annulata. They're great for your garden. They actually are form a bacterial symbiosis. So we got a nice little sample we can kind of take a look at. So there I am liberating the soil sample. From there, actually, I'm going to use this opportunity to like get the sample prepped ahead of time, do a cooking show kind of style. You know, time is always of the essence. Does anyone have any questions while we're doing this real quick? We got maybe like another minute before future me meets up with past me. You guys can unmute if you want.
Yes, I have a question.
Sure.
What's one of your main goals right now?
One of my main goals at the moment in life or with this workshop?
No, with this current experiment.
Oh, so we're going to look at the living fraction of organisms in the soil, as opposed to kind of like drying them and staining them. We're just going to kind of look at them using shadowing microscopy. So that way we kind of introduce you to the cast of characters that are actually in the soil. And then we're going to go from there and introduce further as well. So, all right. So
Craig,
past me is catching up with future me.
Craig, that looked like just potting soil in the on the balcony there, just something from the.
Yeah, there's some potting soil, but also throwing in a bit of compost as well. So,
Okay.
And I'll do some more detail upon the actual sample we'll pull from in general. So we'll go from there. And I'm going to put some of that soil actually out under the dissection scope so we can take a look. All right, so I think I can stop sharing. Cool. All right, so back to my webcam, and then I'm going to share my microscope. So let's see here. So I got my little dissection scope over. Cool, I got my little 1 gram of soil. And we're gonna come to kind of take a look at some of the detail of the soil. So let's kind of look at the soil structure. And we're just gonna kind of do like a rough look, and then we're gonna actually look at the dilution once we focus it in general.
Excuse me, are we looking in the plastic bag now, or did you dump it out on something
Oh,
else?
I dumped out a little section of the plastic bag onto a piece of paper so we can look at it with an actual coin microscope. All right, so we got some structure here. So we see a composition of little bits of wood. Those are the little kind of debris which fungi are actually living in and decomposing. Bacteria are going to be living on smaller chunks of that organic material, the humates, and then the mycorrhizae are going to be binding them all together. So also too, we look at the color, it's kind of like a nice little 70% cocoa brown color. We do see some darker little bits in there. If your soil is black, unless you're living in in the volcanic region like Hawaii, it probably went anaerobic at some point. A lot of municipal compost will cook it. They'll cook it at a high heat to kill most of the pathogens, but they don't amend it by adding in the fungal foods, which refresh the humates in general as well. So we'll kind of use a nice little pick in my dissection kit to kind of push this around. So we'll kind of see a little bit closer structure of, we see all these little kind of channels and valleys or microbiology. And yeah, this is some potting soil, probably some perlite in there as well. But, we can enrich that by working with the microbiology. So anyway, I'm going to switch back over to my main camera. So, all right, so first things first, I got my nice little sample here. I have, a 15 milliliter centrifuge tube, and I've got roughly the sample of soil filled up to roughly 1 mil, 1.5 mil. So we got basically 1.5 grams of soil. So I want to do a 1 to 5 dilution. So I want to add roughly another 4 mils of water. You know, if you want to be, you know, super precise, you can use a little micro, you can use it, wouldn't use a micro pipette, but one of those like plastic little squeeze pipettes. But you know, because I'm a maverick, I'm gonna eyeball it. Cool. All right, and so we got it pretty much on the nose. All right, so we see our sample right there. We see a lot of suspension. We see it starting to settle out. It's a little hard to see because of the glare, but it's starting to settle out. And what we're seeing is the organic matter floating on the top. And then we're seeing the sand, silt, and clay on the bottom, the actual mineral composition actually being there in general. So we've got a 1 to 5 dilution. So we're gonna make our slide. So first things first, let's grab our handy dandy box of microscope slides. Now, when you're getting a slide and you feel like to reuse your slides, rule number 1, you always want to grab one and hold up to the light. Make sure it was cleaned recently. And this one's nice and clear, so we're good. We're gonna put our slide onto our surface. Great. And then we're gonna get our little eyedropper, which we're gonna use to put a little drop of our sample. And we're gonna basically wash it out a couple of times, making sure that there's like nothing dried out, so none of the microorganisms are gonna get stuck to the inside of the actual dropper. All right, so how do we get a good mixture to make sure we get a nice little soil droplet? And what I'll do is I'll switch back over to my little dissection camera. Little microscope slide over there. Cool. And so if you can, so that's all set up. So we looking at me? Okay, cool. So now it's in position. We're gonna take our sample and we're gonna shake it back and forth. Not super hard. We don't want to like centrifuge them out. Imagine if you were hanging on like a, I don't know, like if you're on a lazy river and you're in a log, you wouldn't want to go down like Niagara Falls. You want a nice little, you know, maybe like a jacuzzi tub action. So kind of go back and forth. 1 Mississippi, 2 Mississippi, 3 Mississippi, 4 Mississippi. I'm going to switch hands because I'm right-handed. So I'm going to get my little eyedropper. I'm going to squeeze out the water. Okay, cool. Now I'm going to open her up. Going to go in there, going to get a little drop. Cool. And I've got a nice little suspension of microbes in there, and I want just 1 drop. I'm going to switch my camera. All right, so here we are. Let's do just 1 drop. Okay, so we got a nice little mixture of organic matter, probably some nice little microbes living in there. Not too bad. All right, cool. So next thing, we're going to get our little cover slip. And if I can open it. Hopefully these cover slips don't go flying across my apartment and I got like little shards of glass, you know, kissing me. All right, we got our cover slip. Oh, that one's kind of gross. Let's get a better one. You know, you always got to be careful with these vendors. You never know when the cover slips are clean or they're a little odd or whatever. So there we go. Cool. All right, we got a pretty decent cover slip, so I'm pretty happy with that. All right, then we're going to drop our cover slip on. And the first things first, we're going to want to spread it out. So we're going to kind of want to like move it back and forth. And then we're going to put our cover slip on. So normally when you do a microbe, you like dry it and then you stain them. We want them alive. We actually want to see what's going on. Cool. All right, so we're going to switch cameras to my microscope. And do not be alarmed. I've got a little bit of a test slide on there. So it's, oh, rabbit testicles. I just kind of grabbed it out of a box of prepared slides from Amscope. So that's always fun. All right, cool. So what we're going to do now is we're going to remove that slide, prepared slide. And we have our actual slide we actually want to look at. So first things first, we want to clean our light source. So, you know, whenever you're, so I'm just cleaning my light source. I got a little microfiber cloth. And then we're going to want to clean our lower objective. Divers, that's cool. All right, so that's good. So that'll basically prevent any kind of dust or hair from there on the apartment from like really skewing off what I'm looking at. Cool. All right, so now that's all done. We're gonna get our slide. We're gonna put it on. All right, so now we're going to. I'm gonna try to get some general focus going on. I want to lower the intensity of light. All right, so in the event we don't find anything too interesting, I've got some prepared photos I did the other day, but let's kind of take a little detail in here. I'm gonna just make sure that my intensity of my objective is focused right. So I'm gonna do a little, make sure that all right, 2 yellow, 2 blue, just right. All right. So that way my focus. All right, so let's take a look here. See if everything wiggling around. All right, so let's go from 40x to, let's step it up to, let's go to 100. All right, so we do have some parent material. Got some chunks of sand in there. So those little dots are sand. And this is kind of disappointing. Aw, a little bit of a bummer. Let's a little more light. Let's see if there's anything living on this little bit of humate right here. Let's go to.
What magnification are you using now?
Right now, I am at 400x.
Okay.
I really need to get one of those fancier ones that actually tell you. All right, so we see right here, we see a little bit of the plant matter. You see a little humate right there. You see, okay, so we see some humic clods, a humic substance. We got some bacteria that are floating around. You can see them kind of moving around there. Let's see here, but kind of a bust. So a little disappointing, but usually there's a bit more activity. If also probably would have helped to actually mix the background, but you can see some cocci in there. Got some grains of sand. And what they're doing is those that we're looking at right now is a microaggregate. So it's a little like clod of sand, silt, clay, and humic substance or organic matter, which they're basically making these bacterial glues. But we do have generally good structure at a 1 to 5. But yeah, anyway, so that was a bust, but we only got a little bit of time frame. But in the event of that focus, share screen, basic, cool. In the event of things not going to plan, we're just going to look at some pictures and videos that I had from last time, so you can see actually some living microbes under the soil. And how are we doing for time, Patrick? We got the better part of 8 minutes. Cool. All right, so we got this little video, which will load up in a second. All right, so this was the photo that I showed you before. So we can see here the actual, this is like a really compacted soil. See how it's gyrating and vibrating back and forth? I'm trying to put it on a loop so everyone can see it. But you can see like the bacteria pulsing and moving around between the parent material and the organic material. So this is actually from Manhattan Avenue Park in Greenpoint. So this would be a disturbed soil, ecological disturbance, which bacteria are far more prominent in general. And then so we see here, we see a bit more. We'll see some nematodes, and we'll actually see some strands of fungi along with bacteria. You see a little nematode moving around. He's a little stressed out because he's pressed between the slide and the cover slip and whatever organic material and parent material that's kind of moving there in general. And this one's probably my favorite because we can actually see fungi, we can see a nematode, we can see some plant matter, we can see some bacteria. So we see the fungus, it's moving around that little strand of mycelium. We see the actual net of the plant root. But yeah, so, all right, so what's a slightly easier way to look at organisms that are alive in the soil, right? Soil microscopy takes lots of time. We're synthetic biologists, we're molecular biologists, formally extracting DNA out of the soil. Environmental DNA is great. However, we have no idea whether that fraction of the microbes in your soil, whether they're active, they're dormant, they're dead, maybe it's just, environmental DNA, extracellular DNA. So it's kind of like the same thing of like, oh, I want to look at like what's going on in New York City, right? Okay, let's say I'm an alien scientist, right? And I want to look at, okay, what's like living on the surface of the earth? If I went in and took a core sample that was like, 10 miles wide by 10 miles deep, I'd be looking at a lot of like the geological life, like a stratum over millions of years in general. That's kind of the environmental DNA, even receiving, looking at all the aspects. So this is really exciting. This is from Oak Ridge National Laboratory. So this is using the BondCat system, which is bioorthogonal non-canonical amino acid tagging. So they took a methionine and they modified it. I can't pronounce what the non-canonical amino acid was, but it had an adapter for a fluorescent dye. So they just fed it to the microbes when they incubated them, and then allowed them to tag those microbes that incorporated that amino acid into their proteins and actually see what portion of the microbes are active. So this is really great because Oak Ridge National Laboratory has a nuclear reactor and the soil is really degraded. So it's a nice little effective initial control. And so this is a good way we can actually look at not only the microbes that are active, but at different times of the year, which ones are active, which ones are dormant. And we're actually learning a lot. The Deep Carbon Observatory is realizing a lot of the microbes that are living far deep underneath the surface of the earth, above our initial layer. There's all these biogeomorphologies that are occurring, which we need to understand more about. So that was Seat of the Pants soil microscopy demo. I'm going to basically link to a bunch of other videos along with this presentation and resources back in the Google Drive. But yeah, let's save the last 5 minutes for Q&A. But I hope you all enjoyed. It's great kind of facilitating this at home. I'm glad you enjoyed little bits of it. But yeah, so this is all you need is a compact light microscope that's got an iris diaphragm. And just a couple little tools that are really affordable and accessible. So yeah, feel free to ask any questions.
I had a question I've been stewing on from an earlier breakout session about, how to make people feel like science is important for them. And, how do you engage people with science in a way that then, they can use to help solve their own problems. And and I kind of moshed it together with this workshop, and I was thinking, like for farmers, and I don't know if they already do this, either at the like massive farm scale or like the small family farm level. Could a simplified scaled down version of what you did today be used to surveil the soil to help, farmers make day-to-day decisions like, do I need to change what I'm doing with my crop? Are my bacterial fungal ratios off? Or maybe I'm doing crop rotation and I should do this crop instead of another one?
That's a great question. And the answer is yes. So a lot of the methods that I showed tonight were informed from Dr. Elaine Ingham. So she's done a lot of work to understand how do we understand the dynamics of the microbes in the soil, like in actuality. So this is kind of a component of the kind of regenerative agriculture movement where people are looking at how do we, take care of our soils, understanding that soil is alive. It's actually a macroorganism, a superorganism in general. So it's being used currently as a means to kind of towards regenerative agriculture. A lot of the interest I'm into is how can we take, molecular and synthetic biology, these techniques, and basically, because you're looking at hundreds of fields, you have to do multiple replicates, you have to really have a good data. You have to have all these data points, and it takes a lot of time. You can't really grab a farmer and say, hey, like, let me tell you all this and look through a microscope. You know, so it's kind of exciting because along with the techniques of doing the visual observation, the microscope, the optical microscopy, along with the molecular techniques, we can actually generate these data sets and use the computational power we have to kind of understand a picture about the microbial ecology. Maybe not like a photorealistic painting, but more like a Monet, like water lilies, like the hues of microbial interaction that are going on in general, especially in different times of the year. So yes, currently, and I'll list those resources in the slide folder when I get around to doing them tonight or tomorrow morning.
Hey guys, if you want to turn your camera on, this is the Lax workshop of BioSummit. So let's get some screenshots. Let's get some snaps for Craig. That was amazing and wonderful. Let's all go ahead and say soil microscopy on 3, or mycology.
1,
2,
2, 3. Soil microscopy. Right. So yeah, I'm excited. We're gonna hear from LaDonna, and a big part of what I do is how can we like incorporate traditional ecological principles with the cutting edge science, because a plow, the piece of stick we use to push a furrow, is just as much a piece of biotechnology as a pipette. That's what we leave us with today. So all right,
yep.
see everyone back
Head
in
on over, guys.
That was awesome.